Muy buenas tardes a todos. Bienvenidos al programa de sesiones de Fruit Attraction LiveConnect. En breve arrancaremos con la nueva sesión.
Hello, everyone. My name is Javier Jofre. I am the Corporate Strategy and Marketing Director of Athens. Today and during all these sessions that will take place during the Fruit Attraction LiveConnect event, I will be hosting the webinars. First of all, I would like to thank everyone for joining us today. We have been preparing these sessions with great enthusiasm to continue to share our work and experience in the development of innovative and, most important, effective biological products that provide solutions to the problems that our clients are facing every day. During October, we will be covering different crops of berries today, cereals, and different horticultural crops like, for example, industrial tomatoes, peppers, or artichokes. In our presentations, we will follow with a methodological approach to the use of microorganisms, and we will be ending each session with different experiences obtained by both our own tests and also those of our clients. We will start with berries today, a crop with a high relevance that is being reinforced every year in fruit attraction. To develop this topic, we will have leading the session Mycorrhizae, Technical Director of Athens. Gorka will begin the session with a brief review of our company, continuing with a technical review over Mycorrhiza and Trichoderma, and ending with different success stories in the use of these solutions in strawberry, raspberry, and blueberry. At the end of the session, we will reply any question that you have. Additionally, feel free to contact us at any time by email or using our website. This session and all the sessions will be having during October will be available on the LiveConnect platform if you want to review it later. Without further delay, please welcome Mycorrhizae.
Welcome everybody to this Fruit Attraction LiveConnect webinar. Good morning, good afternoon, good evening, wherever you are connected. My name is Mycorrhizae. I am Technical Director in Athens, and today we will cover some aspects about our company. We will see some tips about Mycorrhiza and Trichoderma, and we will end with a particular application of such microorganisms to berries. We are going to see cases in raspberries, in strawberries, and also blueberries at the end. So let me share my screen with all of you. And please, Javier, do you confirm that you can see my screen? Perfect. So let's start talking about Athens and what we do. Well, Athens is a family-owned company that was founded in 1994. That means we have more than 25 years of history in our bags. And Athens was born to provide mycorrhiza for sustainable agriculture. Nowadays, we are the largest producer of mycorrhiza in vivo system, and we also produce trichoderma in a solid medium. We produce the trichoderma, we'll see that later, in a solid state bioreactor. So Athens was born from the research and development, and it is continuously linked to the R&D. So let me show you also, this is the laboratory of Athens. We call that NGA lab. That means Next Generation Agronomic Laboratory. And we have facilities like this. This is a MySec platform from Illumina. We can take some soil samples and use this machine to know about the populations in the soil. We can discover what are the relationships between the different fungi and bacteria species in our soil. And we also use this machine to know the genetic sequence in our Trichoderma strains. In fact, we were the first company to sequence the genome in our Trichoderma strain. We also have this machine. It's called LCMS. That means like a liquid chromatography linked to mass spectrometry. That is used because from a complex extract from a fruit or from a leaf, we can separate all the components with the liquid chromatography, and we can identify all these components using the mass spectrometry. This is very useful because as a producer of microorganisms, we can also apply these microorganisms to the soil, and then we can see what is the outcome, what is the increase in yield in production, and how the plant grows. But there is a risk there. It's to treat the plant as a black box, that we can make an input and we have an outcome. Okay, the plant can grow well, it can produce better, but if we don't understand what is the mechanism behind this higher yield or higher production, we cannot benefit from the knowledge behind the mechanisms. So we cannot improve our products, and we cannot develop new products that may solve new questions for the agriculture. With the laboratory, in fact, we made a couple of inventions. The first one is related to seed treatment. As a producer of Mycorrhiza and Trichoderma, one of the first questions that arose is that we cannot bind such spores of Mycorrhiza and Trichoderma to the seed surface. Seed surface, it's plenty of waxes, and it's very difficult to coat with Mycorrhiza and Trichoderma spores. So we need to develop a new invent just to discover how that spores can be attached and can be used as a seed coat. So this international patent is also for Spain and at European level, at American and also Brazilian. So we developed this patent just to make our seeds, our Trichoderma and Mycorrhiza spores to coat the seeds. We also developed another patent about reducing water stress in plants. That consists in a plant fiber that can be added to some of our products, particularly the products in tablet format that can be used for transplanting. We add this plant fiber to such products in order to provide a humid ecosystem at the reservoir level, so the plant and the microorganisms can benefit for a higher water availability at this level, and also it prevents from nutrient leaking from the medium. This patent is also at European level, international, American, and Chilean level. So one of the pillars of the Athens is that passion for research, but the other pillar of Athens is the search of the highest quality in our products. In fact, I can show you some pics, and later we can develop that more. But we have some greenhouses to produce our mycorrhiza in our in vivo system. That I will explain you how we can use this system to produce the highest quality mycorrhiza in the market. And to produce the trichoderma, we have a solid state bioreactors. In fact, we have developed it in these more than 25 years. We have developed our own technology, and we have applied the cosmetic industry technology to our aims, and we can mix our products with cosmetic technology, and we can produce our tablets importing technology from the pharmaceutical field. So we can produce all these high quality products, but the farmers also value our products that are zero residue. They have no security periods, and they respect the axonal fauna. And we have a signatory of the Pact for a Circular Economy, and we are focused also on the Food Challenge to 2050, in which we are going to be a growing population in the planet, and we need to produce more in a more sustainable way. In Athens, we have certification for the quality management and the environmental management, also for energy management, and we are fertilizer manufacturers. Our products are for organic certification. They are suitable for organic farming, and all of them have this EcoCert seal. And part of our products are also in the OMRI list from the USDA. But let's start with the mycorrhiza, our first microorganism. Probably all of you know, but I would like to highlight some points about the mycorrhiza. Maybe all of you know that mycorrhiza is a plant symbiosis. It can colonize the plant root, and it can enhance the soil volume that can explore the plant, so it can make the most of the nutrients that we can input. But also it can increase the phosphorus intake. So in some of our soils, the pH is high enough to the phosphorus to be not mobile, and the mycorrhiza can mobilize that phosphorus and can be uptaken by the plant because of the phosphatase in the mycorrhiza. It also improves the water status of the plants, not only because it can explore a larger volume of soil, but also because the fungi hyphae are thinner, 10 times thinner than the plant root. So it can access to the smallest pores in the soil, and that makes a very efficient way to extract the water in case of a drought episode. Mycorrhiza also protects against oxidative stress, and mycorrhizal plants usually exhibit a higher reactive oxygen species scavenge. They enhance the enzymatic activity of the superoxide dismutase and the ascorbate peroxidase, and also they have a higher amount of ascorbic acid. They also have a higher resistance to abiotic stress. That makes the mycorrhizal plants can support better the extreme temperatures, the lower and the higher temperatures, and also the heavy metal contamination, particularly the copper contamination. And they also have a resistance against pathogens. Mycorrhiza is not a nematicide, but as they can enhance the water uptake, if there is a nematode attack in the roots, the plants can make the most of the available water in the soil, and such plants can be less affected. But let's talk about the mycorrhiza in the Athens products. Here we produce 2 strains of mycorrhiza. The products of mycorrhiza in Athens, they belong to these 3 categories: mycorrhiza, mixture of microorganisms, and fertilized microorganisms, according to the Spanish law, the actual Spanish law. The first of the strains that we produce, it's Rhizoglomus irregulari BEG 72. It's one of the most known and studied species in the world. And I know because I'm living in Athens from 2 years, but 10 years ago, I was studying in the university and I was a researcher there, and we were trying to adapt the alfalfa plants to the climate change conditions. And we have a nitrogen alfalfa plant. So the alfalfa was working with nitrogen fixing bacteria, and we wanted to add a third partner. And we were searching the market, what is the more reliable strain of Mycorrhiza, and it was BEG 72 all along. So we decided to start studying with this strain. It's a very efficient strain, and it was isolated in collaboration with the IRTA, that means the Catalan Institute for Research. The other strain is Funeliformes mosaic, BEG 234. It's a different mycorrhiza. It differs from the BEG 72 because it has a largest spore size. And the studies in the scientific literature about the BEG 234 are focused in the large amount of phosphorus they can mobilize, and also the resistance to hydric stress, to water stress. This strain was isolated in collaboration with IFIA, that means the Canary Island Institute for Agronomic Research. So we can have the most efficient strains in the market, but we believe that if you have a good strain, that's just the first step. Then you have to reproduce in the most adequate system that you can afford, just to develop all this potential in the strain. So to produce mycorrhiza, we have different options. The first option that we can think about, it's about to reproduce that in open field conditions. So we have a field with our host plants, we can inoculate with our mycorrhiza strains, and each year we have to take all the soil and then filtrate the soil in order to obtain the spores, which we are going to use to make all the formulations. Well, that's a race. At least two kinds of problems. The first is the irregular production, because each year you may have a different production than the year before. And that, from the industrial point of view, that's a big problem. Also, if there is a pathogen that can be inoculated in the field, it can spread all along the volume of the soil, and then all your production of mycorrhiza is going to be contaminated. So that reason makes us the open field conditions not suitable for mycorrhiza production. In order to limit the pathogen infection, we can opt to reproduce the mycorrhiza in vitro conditions, like a petri dish, like this in the image. We can have half of the petri dish with transformer roots of carrots, usually, and in the other half of the petri dish we have our mycorrhiza, and we can collect all the spores that we want from this side of the petri dish. Okay, we can limit the pathogenic contamination, and we can have a high spore production, but such spores are morphologically very different from the open field condition spores. That makes these spores are smaller and they have less nuclei in the spore, so the infectivity of such spores is very low. If we put such spores to colonize a plant in the open field conditions, it will struggle. So in order to cope with such production systems that has advantage and disadvantage, in Athens we produce our mycorrhiza in a greenhouse. In a greenhouse, we can control that our host plants grow in a substrate with absence of pathogenic plants. We have a high spore production that it is always the same, because the greenhouse remains in the same conditions each year, and the morphology of such spores is similar to the open field spores. So we can ensure that the infectivity of such spores is the maximum, and we will have a high spore life. That means that from the production date to the distribution to the field and the application of the farmer in its field, during all this time, our spores and mycorrhiza are 100% alive. Let me show you in more detail our greenhouse. You can see our house plants growing in tables, not in the soil. And here we have an annual production, and our greenhouse is completely automated. That makes our plants grow each year with the same condition. So we can have the same amount of spores at the same quality each year. And this is industrial, from the point of view of the industria, this is very important. To be in the highest quality and to have a standardized product. We can make, during all this life of the host plants, we can make our own quality controls. And we are sure that our quality controls are correct. But even if this is true, every batch is also analyzed by IRTA as independent center. So we can correlate our quality results with the IRTA to make sure that we don't make any mistakes during the mycorrhiza production. But together with the mycorrhiza in our greenhouse, they're growing also a community of bacteria associated with our mycorrhiza spores. In fact, 2 years ago, we published this paper in a journal in Frontiers in Plant Biology about what kind of bacteria are growing together with our mycorrhiza. In fact, we identify up to 15 bacteria with PGPR properties. That means plant growth promoting bacteria. That means bacteria that can help plants to cope with the stresses and can help plants to grow better in better conditions. Why are these bacteria PGPR? It's because they have nitrogen fixation property, or maybe solubilization of phosphorus, or production of indoleacetic acid. They also can have some production of siderophores. That means a molecule that it can mobilize the iron in the soil, and it can be incorporated to the cytoplasm of the bacteria, and it is also available for the plants. Here there are some photos of the indoleacetic acid production, say the iron mobilization or the phosphorus mobilization. Regarding Trichoderma, it's a very different fungus from Mycorrhiza. Mycorrhiza is an obligate symbiont that makes the Mycorrhiza dependent on the plant to complete its life cycle, but that's not the case for the Trichoderma. Trichoderma is a saprophytic fungus that makes that Trichoderma feeds on organic matter in the soil. So it can be established at the root level and feed on the plant exudates in the roots. But Trichoderma also compete with other fungi for space and nutrients. That when a pathogenic fungi reach to the soil, it can be spread very easily because it competes very well for the space and for the nutrients. If we can add Trichoderma to our soil, it will make very difficult for the pathogenic fungi to make the most of the nutrients in the soil. And also, if Trichoderma established around the roots, it will make very difficult for the pathogenic fungi to reach to the infected sites in the root. So it can rapidly colonize the soil. So we can avoid part of the probabilities to the pathogenic fungi to establish at the root level. It also promotes the defense of the plant through the induced systemic resistance that makes that the plant in contact with the trichoderma, it can enhance the jasmonic acid pathway. So for the plants with a real infection of the pathogenic fungi, the immune system of the plants can react better and more with higher efficacy than the plants with no trichoderma in the roots. It also stimulates the root development through the volatile organic compounds and through the enhancement of the intralacetic acid pathway, and increase the growth of the crop by the synthesis of the auxin, not only the root, but also the rest of the plant, and it produces siderophores in a similar way than the bacteria produce their siderophores. And also, trichoderma can improve the fertility and vitality of the soil. Nowadays, we produce two strains of trichoderma. The first one is a Trichoderma conigy TK7. Is a trichoderma that we actually commercialize. It has a great capacity of the soil condensation, and it comprises all the properties that we have seen before. It has been registered as a fertilizing product in the non-mycorrhizal microorganism group under the Spanish law. So it has a biostimulant registration, the TK7. The other one is the Trichoderma troviridae 8010. That's a different strain of trichoderma, and it's currently in the phytosanitary registration process. In fact, it's in the last phase of approval as an active substance, both in the European Union and the USA. It's a very aggressive strain, and it has a wide range of fungal disease. For example, it's going to be used against Fusarium, Sclerotinia, Botrytis, and ISCA. So let me show you how the trichoderma is reproduced in Athens. This is our solid state bioreactor. It's a vertical 3-module bioreactor. You can see the photo here. You can see the cartoon here for the control display. Here we can select our Trichoderma conigy TK7 strain, for example, and we can make the industrial scale in a completely automated process. As I told you, there are stainless steel modules, and all the process from the cleaning, sterilization, fermentation, and drying, it takes more than 40 days. That means to reach for the best maturity of the spores of trichoderma, it takes 41 days at least. Well, we can ensure that in this fermentator, in this reactor, we have no pollution, and we can maintain the quality, a standard quality that every batch reach. Also, as in the case of Mycorrhiza, in the case of trichoderma, we have our own quality control that can also be assessed by an external quality control. But let's move to the last part of the webinar. That is the reason of the many of you have connected today, and we are going to talk about the berries. What are the benefits for using Mycorrhiza, trichoderma, the combination of both microorganisms in berries. Well, but first of all, let me introduce you to the actual products that contain Mycorrhiza, trichoderma, and we are going to talk about. So the first one is Aegis sim irriga, which is Mycorrhiza in powder format. It has been made for irrigation. It's a powder, as you can see in the picture, that can be dissolved in the fertilization water, and it can be applied to plants in the first stage. It can be distributed through the irrigation system. The composition is 1400 spores per gram, and the dose, it varies from 0.5 to 1 kilogram per hectare. The other product about mycorrhiza, it's called Tim horticola, which is also a mycorrhiza in a powder format, very similar to the Aedes Mycorrhizae, but in this case, the composition it's only 1000 spores per gram. So the dose is 1 kilogram per hectare. Some of the farmers question if these products are really soluble. So we have prepared this video in an actual farm in which one of our collaborators dissolved the mycorrhiza powder product. As you can see, it can be poured into water and it can be dissolved with no problem. Both of the products, Aegis and Tim, they have a granulometry of less than 120 microns. That makes that the product can be passed through all the filters and the drip irrigation system with no problem. You can see later in the video that our collaborator is going to check if there is any insoluble product at the bottom of the container. And as you can check, there is no insoluble parts of the products. So this is the preparation of the broth that can be served through the irrigation system. The other product that we are going to talk about is Condor Shield. Condor Shield is our product with Trichoderma. Trichoderma conigi, TK7, in a concentration of 10 to the power of 9 colony formation units, spores if you prefer, and with Rhizobacteria. It has an organic matter of 56%, and also the granulometry, that's very important, is less than 120 microns. The dose, it depends, 0.5 to 1 kilogram per hectare. It can be higher if it's needed, but this is the standard dose of 1 kilogram per hectare. Both kind of products, Mycorrhiza and Trichoderma, we use them in combination with a third kind of product like this, Heptabiol. Heptabiol, it's a plant extract product. It comes from 7 plant extract, and it says that it has a strong antioxidant activity and a stimulant of the bacteria population. We used to combine Heptabiol with Mycorrhiza because of the rooting formation effect of Heptabiol. The combination of the rooting, and it has also some substance that can make Mycorrhiza to branch better in a faster way, like quercetin. And the combination of the Heptabiol and Mycorrhiza makes the colonization of the roots more easier. So there is no gap, and we can combine Mycorrhiza and Heptabiol to have the benefits of the Mycorrhiza in a shorter period of time. When we combine Heptabiol with Trichoderma, it's to provide Trichoderma a better environment to establish in the soil. Trichoderma, if you remember, it's a saprophytic fungi, so it needs the organic matter in the soil to establish. Unfortunately, some of the soils in Spain, they lack of organic matter. So that's why the mixing of Trichoderma with Heptabiol, which contains total humic extract of 16%, 12% of humic acid, 4% of fulvic acid, that makes a perfect combination with Trichoderma. Well, let's take a look about the success stories. This is for strawberry in the southwest of Spain. Strawberries with no treatment, and strawberries with treatment at the right part of the screen with Aedes mycorrhizae. You can see the line of the plant. It's more covered in the case of the mycorrhiza treated plants, so they can make most of the nutrients that you can input to the strawberries. They can grow faster and they can grow bigger, like in the next picture. In this case, Aedes mycorrhizae treated plants are in the left. You can see that such plants are more leafiness. That means they are growing better. And another characteristic that it's maybe not appreciated in the picture, but it's the uniformity of the plants. The mycorrhiza provides to our fields a more uniform plants, and that makes the manage for the farmer more easier. This is a raspberry growing in pots. You can see how the mycorrhiza promotes the root growing, so it can make the most of the nutrients that you can input, and also it can develop faster and bigger plants that has the potential to reach a higher yield. Raspberry also is cultivated in pots, so it makes a lot of sense to apply a product like this one, Acir Orto, which is a tablet format product. This tablet, which is placed in the transplanting hole or in the pot in case of raspberries, you can put the plant just on the side of the tablet and then cover with the soil. That tablet, as I say, it combines the three of microorganisms. It has Mycorrhiza, 225 spores per tablet. It has Trichoderma, 10 to the power of 6 spores per tablet, and it has the Bacillus Mycobacterium that naturally grows with the in vivo system Mycorrhiza. And it also has NPK. So the idea to combine the three of the microorganisms in the same spot, in the same tablet, is that what we call a consortium, and it's not a random association. We want all these microorganisms to be combined to have a synergistic effect. That means that if Trichoderma improves rooting, that rooting may help to the mycorrhiza colonization of the root to take earlier. And the exudates of these bigger roots can also feed Trichoderma and the bacteria. In this tablet, we also have included the plant fiber that we talked before with the patent, just to provide the microorganisms a good environment, a good humid environment, and can prevent for nutrient leaking. If we can add some NPK in our tablet, we are interested in not to lose such fertilizer in case of abundant irrigation. So let's take a look to the raspberry plants treated with the tablet, with the Acir Orto, and we can see a big difference between the non-treated plants and the plants treated with the tablet. You can see with no doubt that the plants are bigger and they have a very nice looking. In the last years, we have developed a protocol for raspberry cultivation, and it consists on the start of cultivation with Aedes mycorrhizae at 0.5 kilograms per hectare, always combined with heptabiol, 3 liters per hectare, and then after 7 or 10 days, we can add the condor shield, the Trichoderma, 1 kilogram per hectare. And in Spain, we have a first cycle of raspberry and a second cycle. Well, for the second cycle, we can start with the Aedes Mycorrhizae at half the dose, 0.25 kilograms per hectare, combined with heptabiol, 2 liters per hectare. Well, if we look into how this protocol is performed in the plant, this is the difference. This is non-treated plants against treated plants, the right side of the image. So you can see the combination of Mycorrhiza, Trichoderma, and plant extract. It makes more developed roots that can be needed to support a higher production. We cannot have a higher production if we don't develop a well surface of roots that can support all the fruit formation and the fruit filling. So what about the blueberries? Well, blueberry is a very particular plant. It's a plant belonging to the Erbicaceae family. That means that it also established symbiotic association with ericoid mycorrhiza, which are a very particular mycorrhiza. All this time we have been talking about Rhizoglomus and Fundaliformes, and such are endomycorrhiza. For the blueberry do not establish symbiosis with endomycorrhiza. We are currently working in a project about developing mycorrhiza specific for blueberry. But until now, until this is going to be the first product in the market with mycorrhiza for specifically blueberry, until now we do not put mycorrhiza in blueberries. So in April, we start with condor shield, 1 kilogram per hectare, and heptabiol, 3 liters per hectare. This is very important that the condor, the Trichoderma, and heptabiol, they are directed to the root formation, which is one of the main problems in blueberry producers. Then in June, we can add 3 liters per hectare of heptabiol. And then in August, condor shield, Trichoderma, 1 kilogram per hectare. So what is the result of this combination? Well, you can see here in the left image in the picture, the combination of condor and heptabiol results in a bigger plant that has no problems in develop, at least at shoot level, compared to the non-treated plants. What happened at the root level? Well, let's take a look, and we can see here in the right side of the slide, we can see the bigger development of the root. So, problem of the nursery of blueberries solved. We can provide a protocol to farmers that they can use to develop a bigger root system that can support the bigger production. Now I'm going to finish describing a test that was carried out by our colleagues in Chile, from the colleagues from Semillas Ave. They made a test in strawberry. They combined T. morticola, mycorrhiza, and condor shield, Trichoderma, in dose of 1 kilogram per hectare and 0.5 kilogram per hectare. That test was carried out in San Pedro de Melipilla, and it consisted in 2 lines, like in the cartoon here, 2 repetitions of 17 meters length, with a distance between plants of 28 centimeters. So the planting density, the resulting plant density, it's about 55,000 plants per hectare. The plants were shown in May, at the end of May, and in 3 weeks, the Trichoderma mycorrhizae was applied. Well, 3 months later, they remarked that the size of the plants treated with Mycorrhiza and Trichoderma was way bigger than the untreated plants. That peak was taken in September. In November, the plant also grows better, develops better. Here you can have the treated plants and the untreated plants. So what was the result at the beginning of the harvest? In December 12th, they start to measure the harvest, and they were very, very impressed because of the result. So let's take a look to the data of production. So in the first graph, we have the production per treatment in all the 20.4 square meters of that means the line. 6 harvests were performed along the season from December 12 to April 24. And in all these harvests, the treated plants here in orange-red line, they exhibit a higher production. And also, the percentage of the first quality caliber was higher in the treated plants, except for one date, January 13. That was the warmest harvest of the season. Why this low point is explained because of the second quality production that was much higher in the treated plants than the untreated plants. So that percentage was also balanced to the second quality more than the one quality, but not because of the first quality was low, but just because of the second quality berries were higher. So if we see the graphs about the yield, we have seen that the average weight of treatment in such an experimental area was about 6.3, contrasting with the 4.3 of the untreated plants. So if we translate all this data to the yield in kilograms per hectare, that makes that the yield of the treated plants was above 3,000 kilograms per hectare, whereas the untreated plants reach a little bit more than 2,000 kilograms per hectare. And if we look to the average weight of the first quality fruits, we can see that the treated plants reach a higher average, a higher mean than the untreated plants, even if it was not statistically different. But what we want to know with all these data and numbers is, what about the economic yield? Let's take a look at how it results from the point of view of the money to the farmer. So the colleagues in Chile, they create this table in which they classify the price per kilogram of a strawberry from the less optimistic to the most optimistic case in the market. Then they calculate the yield in kilograms per hectare for the untreated plants and the treated with the Athens products, and they calculate the approximate returns in euros well, in fact, they calculated in Chilean pesos, and they have converted it to euros just to make more sense for us. And well, this is the approximate return for the untreated plants and the treated plants. So what is the difference? What is the difference if you have a plant treated with Mycorrhiza and Trichoderma from Athens? And that's the result. This is, as I say you, the less optimistic case, and this is the more optimistic day. Well, if we are pessimistic, we can say that the yield, the average gain of the strawberry price makes that more than 2,800 euros per hectare. Once you have subtract the price of the products. So just to see what are the conclusion of this trial, we have seen that the Athens treatment have shown a greater vegetative development of the plants and a better appearance of health at the end of the season. And the Athens treatment demonstrated a positive effect on the amount of flower set. This is concluded because of the, if you remember, the production of the second quality berries. The second quality berries was way larger in the treated plants compared to untreated plants, and they conclude that it was because of the flower set of such fruits. And the Athens treatment increased the strawberry yield by 13 tons per hectare. What does that mean? Well, that means that the combination between Team Horticola, the Mycorrhiza, and Condor Shield, the Trichoderma, resulted in a 700 profit for the farmer. That makes that in the most pessimistic case, the product is paid more than 7 times. That was very surprising for the farmer. The farmer was very happy, and subsequently our colleagues in Semillas Ave, they were, and as we know that, we are very happy with this result. The product can be paid 7 times with a higher yield reached by such plants. So it only lacks to thank all of you for your attention. We hope you enjoyed the webinar, and if you have any questions, we are here to try to resolve all of them. Javier will explain you how we can contact with all of you. Thank you very much.
Thank you, Gorka, for your presentation. Actually, we already have a few questions. Let me check. We have Ashwin asking about what is the infectivity potential of the Mycorrhiza product per gram, and also is adding what is the reason behind the lower infectivity of the Mycorrhiza product when it's produced in vitro conditions. Maybe you can explain a bit about that.
Yeah, I mentioned it, but yeah, the low infectivity in the in vitro production comes from the well, it's supposed to come from the spore size. Mycorrhiza is it has more. It doesn't have one nuclei per cell. It has they don't have different cells. So the spore, when it's produced, it has several nuclei inside. The effect of the spores with lower size, it makes that the amount of gene material inside is lower, and that is related to the low infectivity of the plants. Somehow, it is not completely clarified until now, but it happens to be.
Okay, thank you, Gorka. We have another question from Harbuk. He's asking about the lifespan, the shelf life of Trichoderma spores and Mycorrhiza products after the bagging process.
Yes, we have checked that in the lab, and we have seen that our Mycorrhiza can have a shelf life of 4 years, maybe 5 in some cases, and the Trichoderma it's more dependent than Mycorrhiza on the storage temperature. But if we take into account normal storage temperature of 25 degrees, it can last between 1 year and 1.5 years.
Okay, thank you, Gorka. And we have one last question from Ashwin again. He's asking about the storage of these products. Specifically, what if the container experiences temperature shocks during the different seasons, especially in tropical weather? He's from India. Maybe he's concerned about the storage of these products.
Yeah, we're talking about Mycorrhiza or Trichoderma?
Mycorrhiza and Trichoderma.
Both Mycorrhiza and Trichoderma. Well, Mycorrhiza it's more resistant to temperature variation. It has more resistant spores than Trichoderma, and even if there are some episodes of high temperature, I mean above 30 degrees, it can be last for quite a long time, like 12 months. But that depends on the way the Trichoderma has been produced.
Okay, thank you, Gorka. Well, I think we don't have any more questions. Okay, we have one last question. It's asking if we have an international patent on the solid state fermentation technology of Trichoderma. Maybe you can explain what patents do we have?
I'm sorry, can you repeat that?
Maybe you can elaborate a bit more about the international patents that we have.
Yes, we have an international patent in the plant fiber to increase the water availability for plant and microorganisms at the root level, and this is one of them, and the other is the patent to bind the Mycorrhiza and Trichoderma spores to the seed surface. Maybe we can prepare a more extensive explanation if he wants, but I mean, I can give the patent numbers if he wants to check that.
Okay, and about the solid state fermentation technology, I understand that we don't have an international patent, but it's own technology, right?
Yeah, it's the technology that we have developed in Athens. I don't know if any other company has such facilities for Trichoderma production.
Okay, thank you, Gorka. Well, with this answer, we close the webinar for today. Remember that we have another one next Tuesday about seed treatment. We will review the Covenant line of products and also the Team Extend seed. Thank you very much and see you next week.
Thank you.